Hello again. So the last part of photosynthesis then is all about compensation points. Now, before we get into this, there's some basics that we need to remember. And the first thing that I want you to try and do is pause the video here and give me the word equations for respiration and photosynthesis. So pause it, see if you can remember them. So hopefully you were able to get these. Now, the equation for respiration is glucose plus oxygen, which reacts to form carbon dioxide and water, whereas photosynthesis is when carbon dioxide and water combine to form glucose and oxygen. And what you'll notice is that the glucose and oxygen, which were produced in photosynthesis, can actually get used in respiration. And likewise, the carbon dioxide and water, which were produced in respiration, can get used in photosynthesis. Now, why is this important? Well, this is important because at certain times of the day, or in certain conditions, in a plant, the rate of photosynthesis will equal the rate of respiration. Now, that basically means that whatever the plant produces in photosynthesis gets used up in respiration, and likewise, whatever it produces in respiration gets used up in photosynthesis. Alpha. And that point is called its compensation point. Oh, Alfie, give over! And that's what this video is all about, the compensation point, and we're going to have a look at some of the graphs involved in it. Now, it's important to understand processes that go on in a plant, and the two main processes are photosynthesis and respiration. So on this side, we can see that when the plant is in light, photosynthesis and respiration both take place. But photosynthesis has two little ticks here, so it shows that it's happening at a faster rate than respiration. At night time, photosynthesis doesn't occur because there is no light, and respiration does occur because respiration has to happen in all living things all the time, or the organism dies. Now, just to show the gas exchange which is taking place when the plant is in light, we've got respiration taking place, which takes oxygen and glucose and produces carbon dioxide and water. So the plant takes in oxygen and it uses the glucose stored in its leaves and produces carbon dioxide and releases it and produces water. Photosynthesis does the opposite. It takes in carbon dioxide and water and produces oxygen and glucose. Now, when the plant is in light, the arrow for photosynthesis is much thicker because it's happening at a much faster rate than respiration. Now, there's pretty much three scenarios a plant can be in. The dark, where there's no light. Dim light, or we can say a low light intensity. Or bright light, where there would be a high light intensity. Now, these three scenarios can actually be experienced on a typical summer's day, let's say. At the dark, that might be at night time. In dim light, that might be dawn, where the sun's rising, or dusk, when the sun's setting. And bright light might be just during the day. So let's look at what processes happen during each scenario. So we've got respiration, and we've got photosynthesis. Now, respiration happens in all living things all the time, so it happens in the dark, low light levels, and high light levels. Photosynthesis will not happen in the dark, but it will happen at low light intensities, and it will happen way faster in high light intensities. So, if you can remember this little table, you can try and remember and compare the rates of each at different scenarios. So, what we can say in the dark is that the rate of respiration is greater than the rate of photosynthesis. In dim light, the rate of respiration equals the rate of photosynthesis. In bright light, the rate of photosynthesis is greater than the rate of respiration. The next thing that we would go on and look at then is gas exchange. So in the dark, only respiration is taking place. So, in fact, maybe we should change this to say that in the dark, it's respiration only. So that means because respiration is taking place and no photosynthesis, that means that oxygen is taken in and CO2 is released. And that's basically just linking together with the equation for respiration. So flick back and look at your equation for respiration now. And then in dim light, because the rate of respiration equals the rate of photosynthesis, any oxygen released in photosynthesis gets used in respiration, and any carbon dioxide released in respiration gets used in photosynthesis. And finally then, in bright light, because the rate of photosynthesis is much greater than the rate of respiration, there is more CO2 taken in during photosynthesis than released in respiration. So in other words, CO2 levels decrease, and as well as that, there is more oxygen released from photosynthesis than used in respiration. So oxygen levels increase. Now from this information, there's one thing that we haven't talked about, which is in here. During periods of low light intensity, where the rate of respiration and the rate of photosynthesis are equal, any oxygen which is released in photosynthesis gets used in respiration, and any carbon dioxide released in respiration gets used in photosynthesis. So overall, we say that there is no net gas exchange. And this is known as the compensation point. So the compensation point is only reached during periods of low light intensity, and it only happens because the rate of respiration equals the rate of photosynthesis. Now this brings us on to the next part. How can you actually find the compensation point of a plant? So you can use hydrogen carbonate indicator, which is this really cool red liquid, and it will change colour dependent on the levels of carbon dioxide. So as I say, it's normally red, and that shows normal levels of carbon dioxide. It will change to yellow if there's more CO2 than normal, and it will change to purple if there is less carbon dioxide than normal. So when there's normal levels of carbon dioxide, that would indicate that it's at the compensation point, so there's no net gas exchange. If it changed to purple, it would show that the rate of photosynthesis was greater than the rate of respiration, because carbon dioxide would be getting used up. If it went yellow, there would be more CO2 than normal, and that would usually happen when there is only respiration happening. So if we look at this example, here we have 5 test tubes, each with different leaves, with different things happening in them. So, using our hydrogen carbonate indicator, we could put a little bit of indicator down at the bottom of each tube. We then put in the different leaves, and we try and find out what happens to them. So, up at the top, or the first one, we have got a green privet leaf. Now you'll notice here that it's covered in black card, so it can get no light. So if there's no light, that means that there will be no photosynthesis happening, but there will be respiration. So overall, it should increase the levels of carbon dioxide, so it should go yellow. The next one has a boiled green privet leaf. Now, because it's been boiled, that means the leaf is dead. So there's no photosynthesis going to happen. And there's going to be no respiration. So in this one, there should be no change. The third one has a green privet leaf. And there's nothing special about it. It's not covered up. It hasn't been boiled. So it should be able to respire, but it will also be able to photosynthesize. And it should be able to photosynthesize at a faster rate than respiration because it's not covered up. So overall, there should be less CO2 than normal. And that's because more carbon dioxide gets used up in photosynthesis than is released in respiration. So it should go purple. The next one is a variegated privet leaf. Now the variegated privet leaf, you might recall from earlier on, has green bits and white bits. Now this will go purple, but will happen much more slowly. And that's because there is less area for it to be able to photosynthesize. So it can respire, it can also photosynthesize, but it's just going to be a little bit slower, so we're just going to draw a little tick. And then finally, we've got a white privet leaf. The white privet leaf has no chlorophyll. So it can respire, but it can't photosynthesize. So it is going to stay red. Now these questions are quite tough, so I think it's important that we actually try and work out what's going on in some of these questions. So an experimental setup is shown in the diagram, so this is really similar to the one we've just looked at. The colour change in the hydrogen carbonate indicator, so it shows that it starts off as red. It will change to yellow if there is high CO2 levels, and it will change to purple if there is low CO2 levels. So it's slightly the opposite of the diagram that we've just looked at, but it's fine. Now we know that it will be red under normal conditions, and it will also show us the compensation point. So what you need to try and do is work out whether or not photosynthesis and respiration is taking place. So what I want you to do is pause it here and see if you can work out what's going on and what you would write in each box. So let's start with A. It's in bright light. The colour of the indicator goes purple after 24 hours. That means that photosynthesis took place. Now, as well as that, it doesn't say the leaf's dead or anything, so it has to be able to respire. In test tube B, it's got a muslin cloth. Now, this muslin allows some light through. So this muslin cloth is replicating low light intensities. So photosynthesis can happen, but it would be at a lower rate. Then test tube C, it's covered in tinfoil. This would allow no light through, so this is replicating the dark. So amount of light received by the light, none. The colour of an indicator after 24 hours, well, because no photosynthesis takes place and respiration takes place, it releases CO2. So therefore, in test tube C, the levels of carbon dioxide would be higher than normal, so it would go yellow. The next wee part is trying to explain these results. So explain why the indicator changed from red to purple in test tube A. So you would use these results to help you out. So in A, photosynthesis takes place at a higher rate than respiration. So CO2 levels decrease as it is used up in photosynthesis. So to answer these questions, you always need to talk about rates, carbon dioxide, and I suppose another good pointer would be the light intensity. So what I should have done for this is said in A, there is bright light, so photosynthesis takes place at a higher rate than respiration, so carbon dioxide levels decrease. So light intensity, rates, carbon dioxide. Now this is a question which is sort of crossing over the respiration and what we've just done. So organisms release energy through respiration. Complete the word equation for respiration. Again, pause it here, see if you can answer it. So glucose plus oxygen produces carbon dioxide plus water plus energy. Give two ways organisms use the energy released in respiration. For movement, growth, for heat. Now in this part, we still use hydrogen carbonate indicator, which we should highlight in red because it is a red indicator. Now this is where you write your knowledge down straight away. So the colours it can be are red, yellow, and purple. So it's going to be red if there's normal levels of CO2, yellow if there's more CO2 than normal, and purple if there is less CO2. Now, in this case, what is the purpose of tube B? Tube B acts as a control. The point of it is to show that any changes which happen in the indicator happen because of the living material. So therefore B has no living material in it, so it acts as a control. Oh, here we are. So, continuing on from this question, hydrogen carbonate indicator changes colour when in contact with air containing different concentrations of carbon dioxide. Yellow if there's more CO2 than normal. Red if there's normal carbon dioxide concentration. And purple if there is air with lower than normal carbon dioxide concentration. So in tube A, the indicator turned yellow after 12 hours. Use this information and the diagram to explain why. So what you would say is in A, respiration takes place. As a result, carbon dioxide levels increase. And then this is where we need to use this information. So there is higher than normal CO2 concentration. So indicator turns yellow. This final one then is an absolute beast of a question. So this diagram shows an experiment to investigate gas exchange. So we've got test tube A, a plant, test tube B, an insect larva, and C, a plant with muslin. So let's look at the conditions in each. So A has a plant. It doesn't have any colouring, so that means it can photosynthesize and it can respire. B has got an insect. Can it photosynthesize? No, because it's an insect. Can it respire? Yes, because it's a living thing. C is a plant with low light levels. So it can photosynthesize, it can respire. Now, what we need to try and do is work out what happens to them. So 3 tubes were placed in a water bath in a well-lit place for 24 hours. The graph shows the results for the carbon dioxide concentration in each tube over the 24 hours. So we need to try and work out which letter goes where. Now, remember, where there is no increase or no change in concentration, that is the compensation point. At that point, the rates of respiration and the rates of photosynthesis have to be equal. So this one is going to be C. This one shows a rise in carbon dioxide concentration. So in that case, there must be more carbon dioxide released than is taken in, which is going to be this one. So that's going to be B. And then finally, this one shows a decrease in carbon dioxide concentration. So carbon dioxide must be getting used up in photosynthesis. So it is A. We then have to show the colour of the hydrogen carbonate indicator in the test tube after 24 hours. So remember, red is normal CO2, yellow is more CO2, and purple is less CO2. So, test tube A, we said that photosynthesis and respiration can take place, so it should go purple, because there will be less CO2 than normal. And that is because photosynthesis happens at a greater rate than respiration. B has an insect. No photosynthesis can take place, only respiration can take place, so it will go yellow. And that's because there's respiration only. C goes red because the rates of respiration equal the rate of photosynthesis. Now use evidence from the graph in the table to explain the results for test tube C. Now remember, the 3 things that we need to talk about here is light, rates, and CO2. So C has the muslin cloth here. It lets some light through. So we can say that there is a low light intensity. Therefore, the rate of photosynthesis is equal to the rate of respiration. Levels of CO2 don't change. Or we can say there is no net gas exchange of CO2. So therefore, it stays red. Another little tricky part. Describe and explain how decreasing the temperature to 15 degrees would change the result in test tube B. So in B, it was 25 degrees C with the insect. Now, if you decrease it to 15 degrees C, decreasing the temperature will decrease the rate of respiration. And that's because respiration and photosynthesis use enzymes, so it decreases the rate of enzyme activity, because enzymes will work at different rates at different temperatures. So the test tube would still go yellow, but it would change more slowly. Now the last question then. So the graph shows the results of investigation into the volume of carbon dioxide used by photosynthesis and the volume of carbon dioxide produced by respiration in a plant between midnight and midday. Now this was a big 6-mark question. Explain how photosynthesis and respiration bring about the changes in the volume of carbon dioxide shown. Now we'll split this into A, B and C between midnight and 5 o'clock. So between here and here. Then at 7 o'clock, which is at this point, and then after 7 o'clock, which is all of this section. So let's get into it then. So A. Now remember, when you're talking about this, talk about light, rates, and carbon dioxide. So link this to respiration and photosynthesis. So between 0 and 5, it's in the dark, so there is no light. So therefore, respiration only happens. There is no photosynthesis. So therefore, there is more carbon dioxide used than produced. B. At 7 AM, there would be low light levels, because that's probably where it's likely to be sunrise. So the rate of respiration equals the rate of photosynthesis. So therefore, any carbon dioxide produced in respiration gets used in photosynthesis. Now remember the other things you say here, there's no net exchange, because that is the compensation point. And finally then, what happens after 7? Light intensity continues to increase. Therefore, the rate of photosynthesis is greater than the rate of respiration. So carbon dioxide, there's more carbon dioxide used than produced. And that's how you would answer that question. Always focus on light, then the rates, then levels of carbon dioxide. And that is the end of this video. Thank you. Goodbye.